All right, before we get started, let's go ahead and do a little review here. So we have E. coli. It's a human pathogen that can use many different types of metabolism depending on its environment. So I would like you to list the types of metabolism in the order of most energy, or ATP, produced to the least amount of energy or ATP produced. Go ahead and pause the video, look over the options, and then I will give you the answer. Okay, the answer would be C. We start with aerobic respiration. That produces the most energy. Then anaerobic respiration is next. And then last would be fermentation. All right, this one is more of a thought question, just checking on your knowledge of structure of bacteria. This probably wouldn't show up on a test. So oxidative phosphorylation, we need an electron transport system, and that's going to develop a proton motive force. In eukaryotes, this happens in the mitochondria between the inner membrane and the intermembrane space, between the two mitochondrial membranes. Bacteria don't have mitochondria, but how do they generate that proton motive force? Where are the protons being moved? So, A, the electron transport system takes place in the plasma membrane of prokaryotes, and the proton motive force is generated between the plasma membrane and the cell wall. B, prokaryotes are unable to do oxidative phosphorylation because they don't have mitochondria. C, electron transport and ATP synthase take place in the cytoplasm of prokaryotes. Or D, prokaryotes do not need an electron transport system. They get ATP from their environment. Okay, so go ahead and think about this one. All right, when I read this, it seems pretty obvious to me. Some of these just don't make any sense, right? We know that prokaryotes do this. We know that they can do oxidative phosphorylation, so instantly two are out. But what I really want you to think about is the answer, which is A. So in prokaryotes, the plasma membrane's there, and then they have the cell wall that's outside of that. So the hydrogen ion gradient is between the membrane and the cell wall. The hydrogens go in there, and then they flow back through ATP synthase. So it's not like they're just going out into the environment. That space between the membrane and the cell wall is actually quite important for bacteria to be able to generate their energy. In 7.5, we are going to talk about carbon dioxide fixation through the process of photosynthesis. So this is a bit of a side trip to the autotrophs. We'll talk about photosynthesis at a very broad level. We're not going to get too into the chemistry here, but it is probably, in my opinion, one of the most important chemical reactions that occurs on Earth. If you're interested in life on Earth, right, this is where all of our oxygen and our fixed carbon is coming from. So we'll talk about photosynthesis. It actually takes light energy and makes a little bit of ATP, and in the process releases O2, and then it uses some of that ATP to fix carbon dioxide into biomass or sugars. And then we'll talk about how photosynthesis can impact human health. Okay, so carbon fixation. I've talked about this process before, but just to reiterate, carbon fixation is taking one CO2 molecule and another CO2 molecule and sticking them together to create a longer carbon-containing molecule. And ultimately, that is how sugars are made. To do that, you need energy to build those bonds that hold the carbons together. And in the process, by a happy accident, oxygen gets made. And we love that oxygen because we need it to breathe. And it does, the oxidative phosphorylation in our electron transport chain. It helps us produce energy. That comes from a process called oxygenic photosynthesis, or photosynthesis that makes oxygen. This comes from photolysis, which is photo, meaning light, and lysis. So light breaking something, in this case, water. So plants need water, as you know, and they take that water and light energy that they absorb from the sun, and they break that water open and produce oxygen. some hydrogen and high energy electrons in this process. Now, remember we talked about oxygen being toxic. It is very reactive, so the plants actually need to get rid of this oxygen. They just excrete it as waste into the atmosphere. And that process has actually changed our planet. Early on in the evolution of life, there was very little oxygen in the atmosphere. But through these photosynthetic microbes releasing oxygen, we have come to have a very oxygen-rich atmosphere. So once we have those electrons, we're going to use them to do a couple of things. We're going to basically use them to capture light energy and make ATP. So this is similar to the process that we just talked about, right? ATP synthase is going to be used, but instead of breaking down sugars to power this process, we're using light. And we start at the bottom here with low energy, and we're going to pump up the molecules to higher energy state and ultimately make ATP. I don't need you to memorize this. I just want you to know that water goes in and we have these electrons. Light hits what we call a photosystem in the chloroplast. That excites the electrons that we've stole from water. They go up to a higher energy state and go through an electron transport system that is going to create a hydrogen ion gradient. Sound familiar? That electron is going to get excited again in a second photosystem by more light. Photons are light. It's even higher energy now, and it's going to go through another electron transport system that's going to generate even more hydrogen ion gradient in this molecule called NADPH. Some of this energy is going to be stored in NADPH. Some of it's going to flow through into ATP synthase. So we have stored energy from light in NADPH and ATP. So we've captured light energy and stored it. Okay, that's the capturing part. Now we're going to use that stored energy to build sugars. So this first part is what we call the light reactions. They are dependent on light. The second part here is independent of light, right? Once we've captured the energy from light, we've stored it in ATP, we've stored it in NADPH. It's then going to get moved over to another cycle called the Calvin cycle, which is actually going to take in CO2 and stick CO2 molecules together, cycling over and over again. We keep adding a carbon, so we start with 1, then 2, then 3, 4, 5, 6, and we end up with glucose as the product at the end. So we're taking energy from light, storing it, using it to build molecules here. Let's go ahead and watch an animation about photosynthesis. It's a little more complicated than we need to know, but I think it illustrates things in motion a little bit better.
Photosynthesis occurs in 2 stages. In the first, called the light-dependent reactions, light is absorbed by chlorophyll molecules in membrane pockets called thylakoids. The absorbed energy splits water, releasing oxygen that forms oxygen gas. The light energy also converts ADP to ATP, and the electrons from water reduce NADP+ to NADPH. These are high-energy molecules that will fuel the second stage of photosynthesis. The second stage is called the light-independent reactions, or the Calvin cycle. Carbon, in the form of carbon dioxide, is taken from the environment and fixed into sugars and other biomass for the cell. The Calvin cycle returns the energy-depleted molecules, NADP+ and ADP, to the light-dependent reactions to become re-energized. In the light-dependent reactions, how does the absorption of light energy allow the cell to convert NADP+ and ADP into high-energy molecules? The complexes participating in the light-dependent reactions are embedded in the thylakoid membranes. The energy absorption includes 2 steps of light absorption, forming a Z-shaped metabolic pathway, the Z pathway of photosynthesis. The membranes contain 2 types of light-absorbing complexes, each containing chlorophyll molecules, called photosystem 1 and photosystem 2. The membranes also contain electron transport systems. Light is absorbed by vast numbers of chlorophyll molecules present in the thylakoid membranes. The absorbed energy is transferred among chlorophyll molecules until it reaches specialized chlorophyll molecules in the reaction center of photosystem II. Here, an electron from chlorophyll becomes energized, which in this diagram is depicted as moving upward. This is called chlorophyll photoexcitation. Notice that the chlorophyll molecule in photosystem II is oxidized because it has lost an electron. This electron is replaced from water by a process called photolysis. Water splits to release electrons, hydrogen ions, and oxygen. An electron from water combines with the chlorophyll in photosystem II, returning it to its reduced state. In a series of redox reactions, the lost electron is transferred from chlorophyll to the electron transport system, and then from one component of the electron transport system to the next. With each transfer, the electron transfers energy to the electron transport system. Photosystem I also absorbs a photon, resulting in chlorophyll oxidation. The electron coming from photosystem II replenishes the lost electron. The final electron acceptor is a molecule of NADP+, which combines with protons and electrons to form NADPH. During its operation, the electron transport system pumps hydrogen ions across the membrane, creating a hydrogen ion gradient. The hydrogen ion gradient stores energy to make ATP. As additional rounds of the Z pathway occur, pairs of oxygen atoms form molecular oxygen. The energy carriers ATP and NADPH are now available to reduce carbon dioxide and assemble sugars through the Calvin cycle.
So there are some photosynthetic microbes that can cause issues for us. In this case study, we have Tonya, who's at the beach, and she spent some time digging up clams. And she noticed that kind of the water near the shore is really clear, but out further, it has sort of a red tint to it. So she cooks up the clams and has dinner, and after dinner, her lips start to feel numb and her fingers are tingling, and she's nauseated, has difficulty walking, speech is slurred, but she manages to call 911, and they come get her, take her to the hospital, and actually have to pump her stomach and give her assistance breathing. The physician's going to explain that she has paralytic shellfish poisoning, which comes from eating clams that have eaten algal toxins. In this case, there's a species that makes this thing called saxitoxin that, as the clams feed on it, they accumulate it in it. It doesn't really hurt the clams, but when you eat them, it hurts you. And cooking doesn't affect the toxin at all. So at the time, Massachusetts had a ban on shellfish because of the high population of this algae called Alexandria tamarense. This can cause what's called red tide, which is the characteristic red water from these algal blooms. This is an interesting one. These algae are actually photoheterotrophs, so they do eat some other organisms as well, but they get some energy from the sun. So they're kind of doing both things at the same time. They are an algae, though, so they're eukaryote. They have flagella and things like that, but they make this toxin called saxitoxin, and it's a neurotoxin because it actually inhibits the sodium channels in our neurons. So it also affects birds, and so it blocks those sodium channels in the neurons, causing paralysis, so your nerves can't fire properly because it screws up the sodium gradient that's there. So this is one way that photosynthetic organisms can affect us. All right, so that's our short bit on photosynthesis. Mainly just want you to know that we're extracting energy from light this time and transferring it to an electron transport system, which makes NADPH and ATP. But that energy is going to be used in the light-independent reactions, the Calvin cycle, to take CO2 and stick them together to build those bonds and to build sugars from that. In this process, this CO2 fixation, as it's called, is building what we call carbon skeletons. So these carbon molecules can be used to build all the other carbon-containing molecules inside organisms. So when you eat these carbon-containing sugars, you can rearrange the carbons and build up new molecules out of them. That's it for 7.5.